All right, let's jump right into this explainer with a bit of a thought experiment. It's a fascinating paradox, actually. Since burning sugar is highly favorable from a thermodynamic standpoint, meaning it inherently wants to release a massive amount of free energy, why doesn't a bag of sugar just instantly turn into energy while it's sitting on your shelf? I mean, that energy is the exact same fuel we use to think, move, taste, and see. Yet a bag of everyday table sugar can literally sit in your pantry for years without spontaneously turning into a puddle of water and gas. Well, the difference between that completely inert bag of sugar on your shelf and the dynamic, hyperactive system of the human body really comes down to one single word: catalysis. You see, without a catalyst, the chemical reactions required to actually sustain life, like oxidizing that sugar, are just way too slow to happen on any useful timescale. We're talking years, if not centuries, for a single reaction. But when you eat that sugar, your body extracts its chemical energy in mere seconds. To get a grip on how biological catalysts, or enzymes, actually pull this off, imagine you're trying to push a giant, heavy boulder over a massive mountain. Now, enzymes don't magically change your starting point, and they don't change your final destination, what we call the reaction equilibrium. Instead, they essentially bore a tunnel straight through the mountain. They create a very specific, highly favorable environment that drops the activation energy you need to get the reaction going. The starting materials and the final products, completely identical. But the enzyme is acting like this incredibly tuned microscopic engine that gets you from point A to point B exponentially faster. So, treating the enzyme like a microscopic engine, here is our roadmap for today. 1. The speed of life. 2. The enzyme substrate complex. 3. Decoding mycolismetin. 4. Limits of the engine. And finally, 5. Throwing a wrench into the system. Okay, part 1. The speed of life. Overcoming the biological crises of slow reactions. You know, for a really long time, the scientific community actually bought into this concept called vitalism. It was basically this idea that biological reactions required some mysterious life force from an intact living cell. But then in 1897, Edward Buchner comes along and discovers that yeast extracts could still ferment sugar into alcohol even when every single living yeast cell was removed. This was an absolute game changer. It proved that fermentation was driven by distinct molecules that just kept right on working all by themselves. That discovery basically killed the idea of vitalism on the spot and kicked off the entire field of modern biochemistry. Fast forward a few decades, and JBS Holding drops a brilliant suggestion that completely shifted how we think about these molecules. He proposed that weak bonding interactions between an enzyme and its target, the substrate, could actually be used to catalyze a chemical reaction. This incredible insight, the idea that non-covalent interactions, things like hydrogen bonds and hydrophobic interactions, are what actually drive the reaction forward, well, that lies right at the very heart of everything we currently understand about enzymatic catalysis. Moving on to part 2, the enzyme substrate complex, the biochemical engine at work. So let's break down how this engine actually fires, step by step. Step 1, you have a free enzyme and its substrate floating around. Step 2, they reversibly snap together to form a temporary, highly transient state called the enzyme substrate, or ES, complex. And this happens in a very specific pocket of the enzyme called the active site. Finally, in step 3, that complex breaks down into the final product. The product floats away, leaving the enzyme completely free, totally unchanged, and instantly ready to catalyze another reaction. Make no mistake, this transient ES complex is the absolute centerpiece of how enzymes work. Which brings us to part 3. Decoding mycolismetin, the mathematical schematic of the enzyme engine. This elegantly simple equation right here, this is basically the core blueprint of our entire engine. The mycolismetin equation calculates the initial reaction velocity, what we call V0, of a one-substrate enzyme-catalyzed reaction. And it was derived using a genuinely brilliant steady-state assumption that was later formalized by Briggs and Haldane. They essentially said, okay, let's assume that during the very initial stages of the reaction, the concentration of that transient ES complex stays totally constant, meaning the exact rate it forms perfectly matches the rate it breaks down. That one assumption is the key that lets us mathematically map the speed limit of life. And mapping it out visually shows us the engine's physical limits perfectly. Think of it like stepping on the gas pedal. At relatively low substrate levels, the speed of the reaction shoots up almost linearly as you add more fuel. But eventually, that rate starts to curve. It begins to plateau, approaching an absolute maximum top speed that we call Vmax. Why does it plateau? Saturation. At high substrate concentrations, literally every single enzyme molecule is occupied and bound to a substrate. The engine is running at absolute full capacity, and pumping in more fuel isn't going to make it run a single microsecond faster. Now, the really crucial piece of this puzzle is understanding the Km, or the Michaelis constant. From a purely mathematical standpoint, Km is the exact substrate concentration you need to get the initial reaction velocity to exactly 1/2 of Vmax. But in practical terms, it's basically your measure of efficiency. If an enzyme has a very low Km, it only needs a tiny trickle of substrate to instantly ramp up to half of its maximum speed. Let's look at part 4, limits of the engine, pushing past the speed limit in the real world. When we actually look at the numbers, these Km values vary wildly depending on the specific job the enzyme is doing inside your body. Take brain hexokinase, for instance. Its Km for D-glucose is just 0.05 millimolar. That means it requires almost 0 glucose to reach half its max speed. It's aggressively efficient at grabbing glucose, which, makes sense because your brain constantly needs energy. But interestingly, that exact same enzyme has a Km of 1.5 for fructose, meaning it is way less efficient at processing that specific sugar. Meanwhile, look at an enzyme like carbonic anhydrase. It has a massive Km of 26. The engine is just tuned incredibly precisely to whatever cellular environment it operates in. Now, if we want to talk about the absolute upper limits of biological engineering, we have to look at the specificity constant. This is a ratio. You take kcat, which is the absolute maximal number of substrate molecules one enzyme can churn through per second, and you divide it by our efficiency measure, Km. And some enzymes, like catalase, fumarase, and acetylcholinesterase, they are so ridiculously efficient that their performance ratio actually hits the physical diffusion-controlled limit. Basically, they catalyze a reaction literally the very instant a substrate molecule happens to bump into them in the cellular fluid. No delay whatsoever. They have achieved absolute, literal catalytic perfection. And that brings us to part 5, throwing a wrench, real-world applications of enzyme kinetics. So trying to analyze that curving hyperbolic plot we talked about earlier, it can be a massive headache. But if we just take the reciprocals of both sides of the Michaelis-Menten equation, we create what's called a Lineweaver-Burke double reciprocal plot. And that bit of math magic visually straightens that complex curve into a nice, clean, readable line. Why is this such a big deal? Because a straight line makes it incredibly easy to diagnose enzyme inhibitors. For example, if you toss in a competitive inhibitor, which is a molecule that literally fights the substrate for the active site, you'll see the slope and the apparent Km change right on the graph. But the Vmax intercept, it stays exactly the same, because if you flood the system with enough substrate, it can eventually overpower the inhibitor completely. And the coolest part is we use these exact kinetic rules to build life-saving drugs today. Take DFMO, for instance. It's a drug used to treat African sleeping sickness. DFMO is what we call a suicide inhibitor, and it acts exactly like a biochemical Trojan horse. It perfectly mimics the natural substrate so it can sneak right into the active site of a target enzyme called ornithine decarboxylase. But once it's inside, instead of a normal reaction happening, it forms this irreversible covalent bond. The enzyme basically uses its own reaction mechanism to permanently disable itself. By truly understanding the underlying kinetics of the engine, we can design the absolute perfect wrench to stop a deadly parasite in its tracks. As we wrap up this explainer, I just want to leave you with one final thought. Think all the way back to that inert bag of sugar sitting on the shelf. The only real difference between that painfully slow chemistry and the rapid, explosive energy that's literally powering your life right now is this intricate system of catalysis. How absolutely mind-blowing is it that a beautifully simple mathematical equation, formulated over a century ago by Michaelis and Menten, physically governs the speed limit of every single heartbeat, every thought, and every biological process keeping you alive today? Thanks so much for joining me, and I really hope this deeper dive into the kinetics of life leaves you wanting to explore even more.
Thank you for watching. See you in the next one. Stay tuned.